All right, our next presenter this morning is Rebecca Grummett, who is a professor in the Department of Horticulture at Michigan State University.
Morning, everyone. I'm really pleased to be able to be a part of this session on genetic resources. And I'll be talking about cucurbit germplasm. So for those of you not familiar with the term cucurbit, that refers to members of the Cucurbitaceae family. But even if you're not familiar with the term, I'm sure you're familiar with these beautiful, flavorful, and often bizarre fruit. So I'll be describing the CuCap project, leveraging applied genomics to increase disease resistance in cucurbit crops. And this is a collaborative, multi-institutional, multidisciplinary project funded by USDA SCRI. We have 19 co-PIs from 10 participating institutions. And the overarching goal of the project is to advance understanding of the genomics of the Cucurbitaceae family and apply genomic tools to practical breeding challenges. So we're working with the 4 major cucurbit crops produced within the US. Those are watermelon, or Citrullus lanatus, melon, Cucumis milo, cucumber, Cucumis sativus, and squashes and pumpkins, which belong to several Cucurbita species. So our first goal then, develop bioinformatic and genomic breeding tools for cucurbits. The cucurbit breeding and genetic community is really small. Across all of these crops, we probably have about 15, maybe public breeders in the entire country. But despite the fact that we have a small community, we do have some advantages. We have diploid genetics and small genome size. And at the time that the first project phase of the project was started in 2015, we already had draft genomes available for 3 of the species: cucumber, melon, and watermelon. And of course, since that time, as for other species, many additional genomes have been assembled, better versions of the ones we already had, the 3 different Cucurbita species, as well as several other species that are of importance in other countries around the world. So the first aspect was to develop genomic databases and bioinformatics tools. And this was done by the bioinformatics team for the project, led by Zhejian Fei at the Boyce Thompson Institute. And so they developed a new cucurbit genomics database with all available genomes and transcriptomes, data functions and data analysis, and this was continually being updated. They also developed some new tools that were originally developed for the cucurbits, but then used by other groups as well, such as a synteny viewer allowing to trace synteny between and among. So this became the worldwide resource for cucurbit genomic data. Now that we're in the second phase, a new version, Reason 2, was recently released, and it now has the latest versions of 33 cucurbit genome assemblies, functional annotations, synteny blocks, and a variety of new functional data analysis tools as well. So a second component of the project then was to genetically characterize the USDA plant introduction collections for the 4 species. So very unfitting with the theme of this morning's discussions. So through molecular genetic characterization then, that can help us to evaluate the extent and nature of variation that exists within the collection to help to manage and preserve diversity and facilitate breeding efforts. And cucurbit breeders have extensively used the germplasm for a variety of traits, including many disease resistances. So what we can see on the figure here is our understanding of the centers of origin and distribution of the crops. So for example, the 2 Cucumis species, cucumber, Cucumis sativus, and melon, Cucumis milo, are thought to have originated in India or Southeast Asia. Watermelon, Citrullus lanatus, from Africa, and the Cucurbita species, the squashes and pumpkins, Pepo, Moschata, and Maxima, from Central and South America. And of course, subsequent to domestication, they were then spread throughout the world. And currently, it's interesting to know that greater than 40% of crop production for all of these crops actually occurs in China. So our goal then was to genotype the collections of the 4 species using genotyping by sequencing, or GBS. So what I'm going to do now is describe what we did for cucumber, because that's the crop that I'm involved with, but parallel work went on for all 4 of these species. So this shows the geographical distribution of the accessions that are present within the US collection. So 1,234 accessions were available to us to be able to perform genotyping by sequencing. From that data, then it was possible to do a variety of different kinds of analyses. And what we see here is the phylogenetic analysis of that collection. And we can see that there are 3 major clades. The first major clade here in green with the deepest branches, that came from India and South Asia, consistent with what our understanding of the center of origin. The second major clade here in orange is the East Asian accessions, primarily China, Japan, and Korea. And then we've got this large third clade, which is essentially the rest of the world. Central and West Asia, Turkey, Europe, Africa, and North America. Now this division into 3 major clades also can be seen both by PCA and k-means analysis, and we see a strong east-west differentiation. And this is consistent with our understanding of the natural history of the crop, so that subsequent to its initial domestication in India, then it moved either east or west and followed distinct trajectories in each case, likely a result of different priorities of what people looked for in their cucumbers, as well as geographical isolation due to the Himalayan mountains. So within the major groupings, we can also see subclades. So for example, we can see subclades within India that corresponded with geographical regions of the Indian states. We can see within the Asian cluster, or clade, that there was tightly clustered accessions from Japan and Korea. They were tight with each other and distinct from the rest of the East Asian accessions. But all of the rest of the East Asian accessions, the vast majority of those were all from China. And it turns out that in China, the North Chinese cucumber germplasm is really quite distinct from the South Chinese cucumber germplasm, and that the Japanese and Korean material is thought to have arisen from a hybridization between the 2. Another interesting observation we can see here in yellow, these are accessions originally collected from Turkey, and they fell into 2 distinct subclades, one that clustered with Central and West Asia, and the other that clustered with accessions from Europe. So if we go to Google Maps and we say, well, where did each of these accessions originally get collected from? Sure enough, those that were clustering with the Central and West Asian accessions came from Eastern Turkey, and those that were clustering with Europe came from Western Turkey, and this fits with our understanding of Turkey as that crossroads between Asia and Europe. Another example we can see in the North American accessions fell into 2 subclades, one that predominantly was composed of fresh market or slicing cucumbers, and the other of pickling cucumbers. And of course, this is consistent with the past almost century of breeding within the US, where these were treated as 2 independent market classes. Another goal of the project then was to develop genetically informed core or diversity panels for each of the 4 species. And so from genocore analysis, we could see that 350 to 400 accessions could capture greater than 95% of the allelic diversity. And so for cucumber, a set of 354 accessions was selected. And in addition, we added historical varieties to include key horticultural traits and disease resistances. And here you can see that the selected group closely mirrors the total population. So what I just described for cucumber was done for all 4 of the crops. So we can see cucumber, watermelon, the squashes and pumpkins, and the melon collection. So in each case, there was approximately 1,000 to 2,000 accessions that were genotyped by sequencing, leading to somewhere between 25,000 and 50,000 biallelic SNPs per species, or 1 SNP for every 5 to 15 kb, approximately 3,000 markers per chromosome. In each case, also, that information was then used to design core populations for the 4 different crops. So moving forward in phase 2 of the project then, with the core collections, our goal is to do deep coverage resequencing, seed multiplication, and phenotyping. And so each crop team either has or is in the process of self-pollinating the selected accessions. Seed companies have volunteered to work with us to provide for seed multiplication, to provide publicly available seed source, and accessions are being resequenced using a combination of Illumina and high-read hi-fi sequencing. Long-read hi-fi sequencing. So the sequence data, the phenotype data, and seeds of the inbred lines will be accessible to the scientific and breeding community. So the first crop group for which we have done the resequencing is cucumber. So the vast majority now resequenced by Illumina at 30 to 40x coverage, and then a handful, or about a dozen, by long-read resequencing. So these data are being used to create genome-wide variome maps, pangenomes, graphical genomes, and to identify SNPs to enable GWAS analysis. Of course, if we're going to make a use of these genomic data, we need to be able to link that to phenotype data. And so the bioinformatics team is currently developing an integrated database module for phenotype, genotype, and germplasm information. The crop teams, in turn, are doing phenotypic analyses of various sorts. So for example, our group is looking at fruit phenotypes in cucumber, both at immature stage and mature stage, and we can, using both photographic and quantitative data for a variety of both internal and external traits. Okay, so the second major aspect of the project is to facilitate, apply these tools to practical breeding challenges. And despite the fact that we're dealing with multiple crops grown throughout the country, a variety of climatic conditions, environmental conditions, consultation with each of the industries identified disease resistance as their highest priority for crop improvement. And of course, this is not really surprising. We know diseases cause severe losses. Control is expensive in both time and money, and I don't think I have to argue strongly to a group of plant breeders that disease-resistant varieties can provide the most cost-effective and environmentally desirable solution. So given in mind then that focus on disease resistance, we turn to each of the commodity groups to say what were your most important diseases that we should be working on in the project. So this just gives an overview for the 4 different crop teams: watermelon, melon, cucumber, and squash, as well as in each case the diseases identified by the commodity groups as their most important. So in each case, each of the teams, the goals are to identify sources of resistance, identify QTL, develop molecular markers, and breed for resistance. We also can use this phylogenetic information to ask some questions about where are the sources of resistance coming from. So for this, we took advantage of disease screening data that was performed by Todd Wayner's group at North Carolina State University. And so for example, if we're looking at gummy stem blight resistance, we can see that there were many tightly clustered accessions. Virtually all came from Japan, a few from South Asia, but resistance was virtually absent from the rest of the world for this disease. If we look at downy mildew resistance, once again, virtually all of the resistance that we observed was from East Asia, again in specific subclades. But we can see that those are distinct subclades than were found for gummy stem blight. And again, virtually absent from other parts of the world. And then looking at anthracnose resistance, we can see numerous accessions clustered in specific subclades in South Asia. Also, we could see scattered accessions from West Asia, Middle East, and Turkey, but again, virtually absent from East Asia in this case, suggesting in this case that the resistances came originally from South Asia and then moved westward. Okay, I'm going to take a moment or two to speak about the cucumber. That's the crop that we're working on in terms of disease, and the 2 diseases that were identified by the commodity groups of most importance were downy mildew, which is primarily a foliar disease, and Phytophthora capsicum fruit rot. And so the downy mildew work is being done by Yixin Wang at the University of Wisconsin, and our group is working with the Phytophthora capsucea. Now, Phytophthora capsucea is a soil-borne oomycete disease, and it turns out for cucumber, this is not the case for necessarily other crops, but for cucumber, it's the fruit that are the most susceptible part of the plant. The leaves and the vines can be pretty much unaffected, they might look great, but the fruit, in contact with the infested soil, or underneath the canopy, it's warm, it's moist, it's a great place for that pathogen to grow. And it also turns out that it's the young fruit that are most highly susceptible. They have a tendency to acquire resistance as they get larger. So this says that all of our screening must be done directly on fruit. We can't, say, spray a bunch of seedlings in the greenhouse. So we've been working with a source of resistance originally identified from the collection from Turkey, lines derived from that. And here we can see that they show a reduced or delayed rate of infection relative to this is a breeding line for pickling cucumber. By 5 days post-inoculation, it's just covered with sporulating mycelia. We also were interested in what's happening in the early stages of infection, so we developed a high-throughput fluorescent bioassay. And here you can see clear differences in growth of the pathogen within 24 to 48 hours post-inoculation, well before you can see any symptom development. A student within the lab then performed bulk segregant QTL-seq analysis, suggesting possible regions of QTL for resistance on chromosomes 5 and 6, and was able to verify the region on chromosome 5 with crosses in both a pickling and a slicing cucumber background. So cucumber, not unlike many crops, exhibits hotspots for disease resistance. So what we're looking at here is a figure from a paper from Yixin Wang's lab, where they put together, at least as of 2020, all of the known QTL for disease resistance in cucumber. And we can see several hotspots for that, and our QTL for Phytophthora fruit rot overlapped with a whole bunch of other in the end of chromosome 5. So it turns out that downy mildew resistance, two of the important QTL there, QTL 5.2 and 5.3, fell within our 4.5 megabase region identified by the initial QTL-seq experiment. And so she went on to develop RILs and use cast marker analysis, and was able to narrow that region to about 0.9 megabases, and fortunately fell right in between the two. And so we're able then to be able to, Yixin Wang's lab is trying to put together all three of them and be able to combine those resistances, and we are currently testing some of those materials in the field this summer. And the other approach being taken then was to phenotype the cucumber core collection for resistance to Phytophthora. You can see variations, some very highly susceptible, others less infected. Those materials have been tested in the field over the past 3 summers, an additional subset being tested this summer to be able to do XP-GWAS. But now that we have just received the resequencing data, we look forward to be able to perform GWAS with the full resequencing data. Now, again, what I've been describing for cucumber has been being done for all of the different crops. These are just some examples showing the fact that we're taking advantage of these genomic tools to identify resistances in various ways, different diseases, different crops. Collectively, then, the team has identified QTL for 22 different crop disease combinations, and molecular markers have been developed and verified for 14 crop disease combinations. And so with that, I want to make sure to acknowledge the people that were involved along the way in the lab for the work I described, as well as the team leaders for the project and all of the other co-PIs for the CuteCap project. And with that, I'd like to say thank you.
We have time for one question for Rebecca.
Hi, Rebecca. Alan Van Dynes. Really nice job explaining the thing. I thought it was rather interesting what you just showed with the downy mildew. You had 2 QTL, but your region of interest is in between the two. So maybe can you comment on maybe what type of populations might be most informative on how to figure that out, or what's really going on, or?
Yeah, I'm assuming that we really are dealing with multiple different kinds of genes in each case. And so I know that Ishwan has been able to now break the linkages between the 2 downy mildew loci and then hopefully successfully slip in the one for Phytophthora.